Welcome back to BioBiz Buzz, the podcast that brings you the latest insights from the biotech and pharma world. I'm your host, Mike Ward, and I'm the Global Head of Life Sciences and Healthcare Thought Leadership at Clarivate, which is a multinational provider of insights, intelligence, and data that the healthcare industry is using to accelerate its innovations. In this episode of BioBiz Buzz, we're diving into one of medicine's most urgent unmet needs, intracerebral hemorrhage stroke. It's a devastating condition that accounts for nearly half of all stroke-related deaths, despite actually representing only 13% of stroke cases. Unlike ischemic stroke, where we've established treatments like the clot-busting drugs, patients suffering from hemorrhagic stroke have been left with a very grim reality. There are simply no approved therapies to prevent the catastrophic secondary brain damage that follows that initial bleeding. The numbers also tell a very sobering story. Globally, ICH kills 3.3 million people annually. That's 45.6% of all stroke deaths. The 30-day mortality rate is approaching 50%, with many of those deaths actually occurring within the first 24 hours. For survivors, fewer than 20% actually achieve functional independence after 6 months. Consequently, this represents one of the most significant treatment gaps in modern neurology. So what if we actually had a medical challenge that came from an unexpected source? Tick saliva. My guest in this episode of BioBiz Buzz is pioneering exactly that approach. Joining me is Marc Dechamps, who is the CEO of Bioxodes, a Belgian biotech company that's developing first-in-class treatment for ICH using compounds that derive from, millions of years of tick evolution. Marc brings over 35 years of pharma industry experience, including leadership roles at GSK, Viv Healthcare, and under his guidance, the company has achieved remarkable clinical milestones, including positive phase 2A interim results that were so promising they stopped patient recruitment early. The company has also secured orphan drug designation from both the FDA and EMA for the lead candidate, which is BioX101. So Marc, welcome to BioBiz Buzz. Marc, let's start with the origin story. What was actually the founding vision of the company, and how did tick saliva research actually lead you to focus on ICH?
That's a good question, Mike. And actually, if you remember, the tick is stinging the host, the skin of the host, not seen by the immune system, so not rejected, but also keeping the blood liquid in order to eat the blood and do the reproductory cycle. So very intriguing, as you can imagine, and the scientists of the Free University of Brussels, ULB, started to analyze what the tick has actually in the saliva, which could generate this situation. Again, not being rejected and not being seen by the immune system and keeping the blood liquid. And after a long research, they identified one sequence of a small protein, 67 amino acids, which is combining, on one hand, an anticoagulant effect, but without increasing the bleeding, and on the other hand, an anti-inflammatory effect, a strong anti-inflammatory effect on the neutrophil, so preventing actually the inflammation or the inflammation in the skin of the host in this case. And with these two properties, we have in this recombinant protein everything which is really, really necessary in order to fight thromboinflammatory indication. And that's the story. At the beginning, the product was developed for cardiovascular indication because the invention with regard to neutrophil and the activity on the neutrophil was not, let's say, identified at the beginning. So just the anticoagulation aspect, but without generating the bleeding. And then at the moment of realized that the action on the neutrophil was so big, so we moved actually the development of the product into thromboinflammatory indications.
Okay, so it started off as an anticoagulant, but you actually recognized that there was potential elsewhere. So can you actually sort of explain the science behind the product that you are now pursuing? And how is it sort of addressing, what's been considered previously an untreatable disease?
Yeah, untreatable disease, as you mentioned at the beginning. So intracerebral hemorrhage, the patient are coming with the primary even to the emergency room of the hospital. So this is the hemorrhage. Unfortunately, afterwards, you describe very well that you have a neuroinflammatory cascade happening, generating secondary brain damages, and nobody and no treatment, nothing could prevent for the moment. By having the ability to prevent the infiltration and the activation of the neutrophil, the release of the neutrophil extracellular traps on the site of the hemorrhage. These traps are very useful when you have an infection, when you have a viral infection or bacterial infection, because they can really trap the virus of the bacteria. But when you have nothing, they are very detrimental, and they start with the neuroinflammatory cascade to unfortunately affect the brain and to unfortunately destroy neurons. On top of that, they are activating the coagulation cascade. So meaning that they activate the factor 12 into 12A. The factor 12A of the coagulation cascade is activating the factor 11 into factor 11A. And then you have the patients moving into a procoagulant state. So meaning that even if you have an intracerebral hemorrhage, you are starting to produce blood clots, which are going to cause ischemia further down the brain. And so you continue to generate damages in the brain. And what is also very important for you to note is factor 12 is also able to continue to stimulate the neutrophils. So meaning it is a kind of deadly loop or vicious circle if you want. And then you have this neuroinflammatory cascade going on. What Biox 101 can do, first of all, as I said, you block the migration and therefore the activation of the neutrophil and the release of the nets. And you block the factor 12 activated and the factor 11 activated, so the intrinsic pathway of the coagulation cascade, meaning that you can stop the formation of the clots, but without generating the bleeding. So we have exactly what we need to have in order to block this thromboinflammatory reaction and to try to prevent as much as possible the damages in the brain of the patients.
Okay, so to sum up, the critical differentiator is that it doesn't cause extra bleeding, like maybe standard stroke treatments, but also it's mitigating the sort of the devastating secondary effects, excessive inflammation and brain damage. That's correct?
That's correct. And you can't give anticoagulant, classical low molecular weight heparin to a patient coming with an hemorrhage. So meaning that today the standard of care is very limited for the neurologist. What can they do? But they are trying to stabilize the patients, decrease the blood pressure if there is an issue, and watch and pray. That's the only thing they can do for the moment. There, they can give an anticoagulant, but without increasing the bleeding, so preventing the secondary ischemia in the brain and blocking the neuroinflammatory cascade by blocking the activation of the neutrophil and the release of the net.
Okay, and the molecule that you've got, its origins are natural. So what are you working with? Is it sort of recombinant?
Yeah, that's a recombinant protein, 67 amino acids we are producing in yeast, GMP production in a well-known CDMO. And this is what we are injecting to the patient. Actually, when the patient are coming to the emergency room, they go to the scan to differentiate whether it is an ischemic or an hemorrhagic stroke. If it is an ischemic stroke, they can do something, as you mentioned. If there is an hemorrhagic stroke, they have nothing to give to the patient. But the patient is in the stroke unit, let's say in the intensive care unit. And so at this moment, at least in the clinical trial, we start to infuse the product for 48 hours in order to cover, with the half-life of the product, 72 hours. Actually, which is more or less the first 3 days during which the people can't give any anticoagulant or anything to the patient to prevent the secondary damages. So we are really covering the moment, critical moment for the patient, but also critical for the physicians where actually they can't give anything.
Well, so I mean, that's clearly impressive potential. So let's turn to the point where you're actually you're translating this promising research into something that could have real outcomes for patients. You've talked about the phase 2 trials that you've been running. Can you give us, where you are with that and the top line results?
Yeah, top line results, because the study is not finished, so we cannot disclose all the results without having a confidentiality agreement. So we decided to go for a phase 2A after the phase 1 for sure. In a phase 2A. Phase 2A, why? Because we are giving the product for 48 hours. In the phase 1, it was for cardiovascular indication. We were giving the product for 6 hours. So we had to redo all the PK/PD analysis for the regulatory authorities, which is normal. And we started an open study, but randomized study, looking first for safety and tolerability, because we are giving an anticoagulant to patients coming to the hospital with an hemorrhage. And also to start a secondary endpoint to look at biomarker and some signals with regard to potential efficacy. Actually, we stopped the recruitment after 23 patients because at the time we realized that we met already the primary and the secondary endpoint. So primary endpoint, safe. The product is totally safe. The only thing we can see, we have just one patient with a reaction at the site of the injection, a little bit like for a vaccine. So it is really nothing. So the product is extremely safe. From a pharmacokinetic and pharmacodynamic perspective, it's spot on, exactly what we expected with the model we have, meaning that we cover the patient during 48 hours, but we cover in reality the patient for the first 3 days with the duration of the activity of the product. We block the factor 11A and 12A at the level expected. We have an APTT ratio, which is measuring the anticoagulation effect exactly at the level which was expected. And next to that, what we saw is a decrease of the hemorrhage volume. We expected to have a stabilization, but we see a decrease. And we see also a decrease in edema formation around the hemorrhage. Edema is definitely what you can see with MRI and scan, and essentially MRI. And this is the signal of the neuroinflammatory cascade around the hemorrhage. So next to that, there is a biomarker called neutrophil to lymphocyte ratio in the blood. And when you see in the blood that the neutrophils are very, very high compared to the lymphocyte, this is really, unfortunately, a marker showing that the patient will have potential mortality or very big difficulty to recover. And we see that with the standard of care in the blood of the patients. But what we see actually with the patient treated today, but again, it's experimental. We just have 23 patients. We cannot draw any statistical conclusion, but we see that we have a flat curve with regard to the neutrophil, meaning that we can really block the migration and the activation of the neutrophil. So next to that, we are also looking for a functional recovery for the patient. Again, that's experimental at that level with the 23 patients, but we see at least at day 90 that we have a better recovery, at least better signal of recovery compared to the standard of care as well. So we are actually ticking all the cases we wanted to tick. And that's the reason why now we are really looking forward to move into a bigger trial, an efficacy trial for the development of the product.
Yeah, I mean, can you share with us sort of any sort of feedback you've had from key opinion leaders or sort of clinicians in this space?
Clinicians and key opinion leaders. First, if you look to the people in the centers where they used actually Bioxodes in the clinical trial, they were considering that the patient was recovering better, but it was a soft feedback. Now we see actually, again, it's a signal, it's a trend, but we see that it is translated into a MRS score. Next to that, we have a very strong clinical advisory board, international advisory board, with people very specialized in the stroke, in the hemorrhagic stroke in particular. And they're happy to eventually have something to give to the patient, because just realize that you are a treating physician, the patient is coming for an intracerebral hemorrhage, and you need to contemplate the patient and hope that the damage will not be that much for the patient, that he or she could recover at least partially. Now, what they see is a signal that they could move forward into a next step of clinical trials and hopefully generate new hope for the patient and new hope for them to eventually have the possibility to prevent the secondary damages. So definitely, we have a very spontaneous and very positive support of key opinion leaders for the development of Bioxodes in this indication.
Yeah, I mean, it's clear why they would be enthusiastic. Biotechs, though, developing innovative medicines for those unmet medical needs, it's a common target for biotech. And there are often really, really good reasons why a condition has remained untreatable. So what unique challenges have you faced developing a treatment for a disease where currently no therapies actually exist?
I would say two challenges. The first challenge is to develop a protocol, because there is no specific protocol for intracerebral hemorrhage. So really to work hard with the clinical advisory board to define the endpoints for the phase 2A. We are actively working with them now to define the future protocol of the phase 2B. We are also discussing with the authorities, FDA, EMA, really to consult in order to fine-tune this protocol synopsis for the next stage. But again, because there is no real experience, because all the treatment failed in the past, and the treatment of the trials were based on ischemic stroke and not specific to intracerebral hemorrhage. So really to create from scratch what will be the right protocol to move forward, and hopefully to bring this product to the patient with a regulatory approval. The second challenge is definitely the funding. Let's be clear, let's be honest. It is considered by the VCs and by the strategic, by the pharma companies, as a graveyard of investment, because nothing happened in that field. And even in ischemic stroke, it's not that great. In intracerebral hemorrhage, in hemorrhagic stroke, there is nothing. And so everybody was asking, fine, guys, go back to us at the time you will have clinical data. So for us, it was really about being very resilient, working with our current shareholders. Most of them are public investors, high net worth individuals, family office people. And I would like really to thank all of them for the support we received, because thanks to them, we were able to produce this data. Now, we are discussing with VCs, we are discussing with pharma industry, and we want to move forward. But to your point, this is a challenge we have in front of us. We need to raise now 70 to 80 million euros in order to move forward with the phase 2B. Also having in mind that we receive orphan drug designation in the US and Europe. So because there is nothing, to your point, there is nothing to treat the patient, we might end up with the next clinical trial, with the pivotal trial, meaning that we could potentially with a partner, pharma partner, have a conditional marketing approval and having the possibility after the next clinical stage to bring this product to patients. So you see the challenge. Challenge is to have the right protocol with the right endpoint, not only for the medical community, but also for the regulatory authorities to approve, even conditionally, the product, and also for the VCs and potential pharma partners for really getting the fund for moving forward with an international trial.
So, when it comes to potential pharma partners, so is your business model to actually, it would be a pharmaceutical partner that would eventually launch that product into the marketplace?
You mentioned that I have a strong background in the pharma industry and in GSK, Vivelcare in particular. So, and I did commercial activities and company management during my pharma life. So I know what it is. And at Bioxodes, we are just 7 to 8 people. We don't have the infrastructure actually to go for commercialization. So the preferred business model is really to find the right partner and support the development today to give the partner the right product for commercialization all across the globe. Really, the intention of the team is really to bring this product to the patients who will need Bioxodes 1 to prevent the secondary damages and in the best way forward and as quick as possible for the treatment of the patients.
So that's interesting, because one of the things that I've seen recently, it's a sort of news coming out of Varmex, which is a biotech company that was spun out of the Leiden University Medical Center based in the Netherlands. And they entered a partnership with the pharmaceutical company CSL. And it's basically to develop Varmex's phase 3 drug candidate, VMX-C001, which is there to sort of restore coagulation, to stem bleeding in patients who either need urgent surgery or who are using anticoagulants. I mean, that deal it's impressive, $117 million up front, and you're potentially $2 billion in fees for acquisition and contingency value rights in exchange for an exclusive option to actually acquire Varmex. So I was just sort of wondering, in that sort of context, can you sort of describe what the sort of the potential read across for Bioxodes actually might be from such a deal?
But this is a good news for Bioxodes. This is a good news for the field. Huh? So it really demonstrates that pharma industry is again interested in that sort of development. Huh? Because if you look to what they would like to acquire, it is something, to your point, which is restoring coagulation. We are also in the anticoagulation space, obviously with a totally different mechanism of action and outcome. But Varmix is also targeting a mechanism closely associated with a mode of action because this is the intrinsic pathway. So the goal for them is to prevent, let's say, bleeding when a patient is coming to the hospital for surgery, for instance, with a risk of bleeding, which is important. So you have to stop anticoagulation immediately. For us, if you remember, we are stimulating an anticoagulation situation for the patient, but without increasing the bleeding, which is totally different. And on top of that, we have this very strong anti-inflammatory effect on the neutrophil. But nevertheless, this is really good news for us. On one hand, pharma is again interested in that field. On the other hand, investors have seen this deal last week, so meaning that they might be much more open for this type of discussion. But again, the differentiation between Varmix and us, even so we are more or less in the same area, is definitely that we have a potential for preventing secondary damage in the brain of the patient for stroke, but also for all thromboinflammatory indication.
Okay, yeah, so that's really interesting.
Very interesting for us, and obviously a good benchmark for us as well. So looking forward to go for the same type of deal in the coming years. Yeah.
I mean, what we're seeing also these days, whether it's with respect to sort of your VC investments or even pharmaceutical partners, that they're looking actually for not just interesting products, but actually products that could potentially turn into platforms. So are you already sort of thinking that, what you have with, say, for example, Biox 101, that actually has potential for other indications?
Definitely potential for other indication, and the next indication could be ischemic stroke, because when you do thrombectomy or thrombolysis, unfortunately, not all the patients are having, let's say, a better improvement because you might have difficulty for reperfusion and so on. And what we see, it is exactly the same mechanism. We find clots based on neutrophil, on nets, everything related also to the secondary damage of intracerebral hemorrhage. So just imagine that, because now we know at least for 17 out of the 23 patients that we are safe. But if we confirm that we are safe, if we confirm that we are effective in intracerebral hemorrhage, we can move forward directly almost in phase 2 in ischemic stroke. And at the end of the day, the people treating stroke patients could be in a position to give immediately Biox 101 to prevent all those damages. And having Biox 101 for, let's say, stroke patients in a general term. That's the long-term vision. But next to that, there are other thromboinflammatory disease, unfortunately today without any solution. Because you can look to the literature and you can look to what companies are developing for the moment, big pharma or biotech. At least publicly, we don't see any other companies developing something in that field. And we don't see any results compared to what we are starting to see with regard to neutrophil and so on. And next to the other thromboinflammatory disease, I cannot disclose here because it's still confidential, we want to continue to the discovery program specifically on the neutrophil. Because there are a lot of inflammatory disease mediated by the neutrophil, chronic acute situation with high unmet medical need. Just imagine that we could use just a subset of the Biox 101, which is responsible for the activity on the neutrophil, and develop a new entity, whether it would be a recombinant protein or another formulation, but having the ability to treat acute but also chronic inflammatory disease in that setting. So we could develop a new platform on top of the one we have developed based on the tick saliva.
I mean, this has been really, really fascinating. So as we bring the conversation to a close, could you sort of, in a nutshell, sort of describe what you think success will look like for Bioxodes and, I guess, ICH treatment landscape in the sort of the next 5 years or so?
Yeah, but definitely, yeah. When you discuss with the key opinion leaders, when you discuss with physicians, bringing at the end of the day something to the patient to prevent secondary damages. And we don't speak about the population, we speak about the population unfortunately suffering from intracerebral hemorrhage in general, would be a miracle compared to what they have for the moment. So already it will be the big success of the company, because we could be the first company bringing something in that area and something really for the patient. And next to that, for our shareholders, having the ability to really, at the end of the day, seeing a pharma company is taking the program and bringing the program to commercialization and starting to treat the patient all across the globe would be another big success for the company. Next to that, the development, to your point, of new indication and a new platform, because we still have quite a lot of work to do.
Yeah, Marc, thanks very much for sharing this remarkable journey, from tick saliva research to a potentially life-saving therapy. The progress that you've just described in this sort of, clearly what is a critical unmet medical need is truly inspiring. So we'll be following those phase 2B developments very, very closely. For our listeners, this is exactly the kind of innovation that demonstrates how biotech companies are actually tackling medicine's biggest challenges through what might appear very unconventional approaches. So from all of us at BioBizBuzz, best of luck, Marc, with your Series B and the road ahead. And that's all for today's episode. I'm Mike Ward. I'd like to thank Marc for joining us today. And I also hope that you'll tune in next time for the next edition of BioBizBuzz. Keep innovating, and goodbye.
Thank you, Mike.